So yeah, welcome to a Systems Neuroscience and Complexity seminar today. Really excited to have Chris Whyte, or Christopher Whyte, as we're now desperately trying to call him. I hasten to use the word brilliant, but I think it fits in this case. Christopher, grad student in my lab, that's been keeping all of us on our toes for the last little while, and really excited to hear what he's up to. He's been sort of trying to combine neurobiology and dynamical systems, but in a way that makes contact to cognitive, perceptual, attentional phenomena. And so I think it's a really audacious, but I think tangible research project that we're really excited to see evolving in real time. We're all learning, I think, as we go, how to do this in the right way, where the pitfalls are, what the false starts and the profitable ways forward are. And I'm really excited to be on that journey with you, Chris. So excited to hear what you're up to today, and take it away.
Everyone see this? Hopefully you can. Okay. So which one is everyone seeing currently?
This is the presenter view. You had it on the...
Yeah, so I'll swap displays. How about now?
You got it now, yeah.
Got it now? Nice. Okay. So yeah, just to say return some of the compliments. I think when I first met Mac in 2019, the thing that most struck me wasn't necessarily, it was really kind of a very unique way that he was going after kind of being a scientist, where kind of this novel integration of neurobiological theory, computational modeling, but also just kind of like being really deeply curious about just how things work and using whatever data set fits. And that kind of left a really lasting impression on me and kind of showed me how I wanted to do science, I think. And so the last year has just been really kind of fantastic. So thank you. Okay, with the bromancing out of the way, let's get started. So this is probably going to be about a 35-minute talk. I think it's pretty rare that a grad student kind of gets the chance to be a little bit self-indulgent and kind of really dive into a project at least and get kind of this level of feedback. So I really want to take advantage of that. So today I'm going to be talking about some recent work from our group where we try and explicitly link the essentially the cellular mechanisms of perception that are typically studied in rodent models to the kind of behavioral signatures of conscious perception that are typically studied in human cognitive neuroscience. So we're going to be doing this using kind of, as our example paradigm, basically visual rivalry, which is a visual phenomenon I'll go into in a moment's time. Okay, so whoops. All of this comes from basically a recent preprint from our group. So if any of you are interested, hit it up, we can get a copy and chat about it. Okay, so the background to all of this is really in my interest in the neuroscience of consciousness, which I take to, hopefully it's an uncontroversial thing to say, that largely this is basically all human neuroimaging or human and non-human primate neuroimaging. So on the right, I'm showing two fantastic studies. The first is a meta-analysis that basically looks at the neural correlates of conscious or visual awareness across paradigms. And they basically find this contrast that distinguishes between aware states. So this might be a stimulus that you can report and a stimulus that you can't report. And they find this frontoparietal network that distinguishes between these two, between this contrast. And below is basically a study looking at essentially the same paradigm or the same idea, but in basically a case of intransitive consciousness. So this is whether you're awake or under a general anesthetic. And they put macaques under a variety of anesthetics and look at this contrast. And so global neural workspace theory, integrated information theory, higher order theory, recurrent processing theory, they're all macro anatomical theories in the sense that this is the type of data that they're trying to explain, right? And I think, like, power to them. I think they've done actually a really remarkable job despite like some recent controversies. I think the field is making really good progress and it's really healthy. But with that said, I do think that this is kind of the modality that we've paid attention to has almost exclusively been non-invasive neuroimaging. And this has basically shifted our attention away from kind of neurobiological detail and mechanism toward these large-scale networks. And so I think if you read these kind of the early papers from global workspace theory, integrated information theory, recurrent processing theory, they all have these cellular level commitments as kind of like deep explanatory mechanisms in their theory. Global neural workspace theory talks about layer 2-3 cells, and basically the really broad connections of these being really important for global broadcasting. Integrated information theory talks about the grid-like structure of V1. But basically these these aspects of the theory are really unexplored. And it's not kind of anyone's fault, really. It's just really hard. And so when I joined Mac's group, I became aware of this really kind of stunning set of results from Matthew Larkham's group in Berlin, where they're essentially going after just the same questions as everyone in kind of typical kind of cognitive neuroscience of consciousness, but they're doing it in rodent models. And they're really interested in this particular circuit, thalamocortical circuit, connecting layer 5B pyramidal neurons, which have their soma in layer 5B and their apical dendrites in layer 1. And they are essentially, this is also, essentially, they are essentially the nexus of feed-forward and feedback information flow in the brain. Feed-forward input hits the soma in layer 5B, feedback input hits apical dendrites in layer 1, and they're also basically the primary output of the cortex to the subcortex, and they form this kind of arguably closed-loop connection with matrix thalamus. And what they show over a variety of different kind of experimental setups was basically that the integrity of this circuit, I'm going to go into the integrity of what I mean by integrity of the circuit in a moment's time. This is a crucial necessary condition for both kind of conscious level, being awake versus under a general anesthetic, and actively perceiving a stimulus versus a stimulus being in the nervous system, being propagated, you could decode it, but not perceiving it. And I'm going to focus really on the perceptual side of this today, but I mean, Eli, you've talked about some of this work in your recent snack, and Brandon's done a whole bunch of work on this too. Okay. So what's so crucial about this cell, and what's one of the things that's so interesting, is that when it receives coincident input into the soma and the apical dendrites, basically it will display this dramatic change in its dynamics from this regular periodic spiking behavior, which is kind of typical of pyramidal neurons throughout the cortex. In particular, layer 2-3 is kind of a canonical example of this. And so when it receives just feed-forward input, it's just that periodic spiking behavior. When it receives just feedback input, there'll be some depolarization of the apical dendrites, but there won't be a calcium spike, or there'll very rarely be a calcium spike, let alone a sodium spike in the soma. But when there's this coincident drive into the soma and the apical dendrites, you get this remarkable switch in the dynamics of these cells from this regular periodic spiking to this bursting regime, where it triggers a sodium spike. That sodium spike will head up the trunk of the cell. This will then basically depolarize the apical dendrites to a sufficient degree to trigger a calcium spike, which will then propagate back down and basically trigger this bursting where the cell essentially doubles its firing rate. And what's interesting about this from kind of perspective of conscious perception is that this bursting seems like a necessary condition, or at least time-locked bursting seems like a necessary condition for conscious perception. So this is this really lovely study, again, from Larkham's group, where they basically have a threshold detection paradigm where they have this kind of stepwise deflection of the whiskers that they basically titrated to the animal's perceptual threshold, and they have it report when it perceives the stimulus or when they think it's there. And on trials where it reports hitting it, so basically a hit trial, you'll see this time-locked bursting, which are these green dots in the raster plots. Whereas on misses, there's basically fairly asynchronous bursting just because the animal's in like an awake state. But what's really crucial about this is that you can also basically control the animal's perceptual threshold by controlling the extent to which these cells are able to burst. So if you pharmacologically inhibit the apical dendrite through the application of baclofen, which is a GABA B agonist, you can essentially shift the psychometric function right. You're increasing the perceptual threshold. And you can also do the opposite by optogenetically exciting these apical dendrites. And so when you do that, basically the probability of reporting a threshold level stimulus shoots through the roof, but also the false alarm rate shoots through the roof. And I think kind of a really daring, but also very plausible explanation of what's going on there is the animal's hallucinating when you stimulate its dendrites. And crucially, you can also essentially get the same thing as kind of this pharmacological inhibition of the apical dendrites by pharmacologically inhibiting the downstream targets. So here I'm showing results from pharmacological inhibition of the posterior medial nucleus in the thalamus and the superior colliculus. Posterior medial nucleus is like matrix rich, and the superior colliculus will project back up through the matrix thalamus again to these layer 1 dendrites. And so all of these things will basically promote bursting. Okay. So the goal of this work I'm going to show you today is to really see whether these findings in rodents, that layer 5B bursting essentially seems to be a determinant of perception, can explain anything, any of the kind of rich set of perceptual phenomena that people in cognitive neuroscience, perceptual psychology are kind of interested in. And it's not clear to me that it will, or at least going into this, it wasn't clear to me that it would. It's a pretty idiosyncratic perceptual paradigm, right? Where they're just like deflecting these animals' whiskers. But at the same time, like theories of visual awareness, at least in my opinion, in humans, they lack any kind of like real neurobiological basis, at least at the level of kind of cellular detail that's in a lot of this rodent work. And so I really think there's a lot of room for progress to be made by building models like explicitly link these cellular level mechanisms studied in rodents to the kind of mechanisms that are studied in human cognitive neuroscience. And doing this through basically studying tasks can be done in both. And this kind of parallels basically the emerging consensus in the field. So this is a recent kind of meeting report from Bai He, where she summarizes basically the points of consensus in a recent meeting at the NIH among like a pretty heterodox group of people. I think Stan DeHaan and both Julia Tononi were there. And so these are not people that agree about much, but they all kind of converged on this idea that the approach needs to be kind of neurobiologically grounded and relatively theory neutral. And we also, crucially, need to be able to study consciousness in parallel in both humans and non-human animals. And for this to work, we need to basically rely on non-verbal reports. You can't ask a mouse to like provide a detailed, like subjective report of its visual experience, right? It has to be some kind of behavioral readout. And so really, with these kind of goals in mind, we went into the consciousness literature and the perceptual psychology literature looking for a task that was basically satisfied this criteria. And what we arrived at was binocular rivalry. And binocular rivalry is great for a number of reasons. One, because it's incredibly robust. And two, because you can do it in humans and non-human primates. It's been studied for decades in humans and non-human primates. And crucially, because of recent advances in basically experimental paradigms, you can now study it in rodents. And there's been 2 studies in rodents. I'm going to talk about one of them in a minute. And for those people who kind of aren't aware, binocular rivalry is this visual phenomenon where if you get incongruent input to both eyes, so here it's orthogonal gratings of different colors, but could be a face in a house, could be a variety of things. Instead of seeing some odd superposition of the two, what you get is these fairly discrete alternations between the input from either eye, with brief periods of mixing between. And if you're interested in kind of the neural mechanisms of perception or conscious perception, this is great, right? Because we have 2 physically mapped stimuli. They control for basically all of the physical aspects of these stimuli. In one case, it's perceived. In the other case, it's not perceived. But the information about both of these stimuli is in the nervous system. Okay. So basically, we took as our parts list for the model, it was just this recurrent circuit connecting layer 5B pyramidal neurons to matrix thalamus. And we also added in an inhibitory neuron, basically to stop runaway activity. And so as our model neuron, we chose Zikovich neurons. And these are these quadratic integrate and fire neurons that are with a adaptation current in there, that are incredibly computationally efficient, but also with the right parameters are capable of reproducing Hodgkin-Huxley style dynamics. And so they've also, I mean, fortunately for us, been pre-fit to all of the cells we're interested in, including these layer 5 bursty neurons. And so we basically built a model with this as our parts list, where our layer 5 PT type neuron is actually a dual compartment neuron. So we have a separate compartment with each compartment consisting of a Zikovich neuron for the apical and somatic compartments. And then we coupled them together such that the dynamic regime of the soma depended upon spiking activity in the apical dendrites. And this was an insight that Brandon had, where basically what he realized was that the difference between regular spiking and bursting in terms of parameters, the only difference is actually in terms of the reset conditions on the soma. And so you can basically reset the point at which these neurons are reset on the vector field in terms of basically the baseline membrane potential and the amount of spike time adaptation that each spike contributes. If you reset that, or if you make that contingent upon whether there's spiking activity, you can very efficiently reproduce what would otherwise require a fairly complicated multi-compartment model. And then, of course, we also took parameters for basket cells and thalamic cells for our matrix population. And really the thing that we wanted to kind of make sure that we could qualitatively reproduce was this finding from Matthew Larkin's group, where if you increase kind of apical drive, so the amount of spike activity in the apical compartment, this will effectively control the gain of the somatic compartment. So let's say I pick, say, 400 hertz of basal drives. This is a Poisson process driving my somatic compartment. With no apical input, we'll get about 20 hertz. If I put in 300 hertz of apical input, that's really about the max what we put in. We essentially double the firing rate. And that was something that's basically found in vitro. Okay, so basically we then took, with our kind of model neurons in hand, we then coupled them together on this cortical ring with these kind of bi-exponential conductance-based synapses with prefit parameters for AMPA, GABA, and NMDA receptors. And so our cortical ring was basically consisted of the cortical component of our model. It's these layer 5 PT type cells and the corresponding basket cells which provide EI balance. These then have recurrent connections with a kind of like matrix thalamus ring. And so basically each 10 of the neurons on our ring, on our cortical ring, project down to one matrix thalamus neuron, which then projects back up diffusely to 10 cortical rings. Okay, and so crucially, because both in the brain and in our model, inhibitory connectivity is broader than excitatory connectivity. When we drive it at opposite ends of the ring, basically with a Poisson process, in two independent Poisson processes representing input from HI, and then weighted by a spatial Gaussian, basically representing orthogonal grating stimuli that's typical in binocular rivalry. What you get is this like basically mutual competition for dominance. And so what I'm showing you here is basically just like a quick animation of some basic operation of the model. The first is essentially a raster plot of the full 90 cortical neurons, of the excitatory neurons. And below is basically a model neuron from basically the center of each of these competing populations. So what you see initially is this kind of flurry of activity as the stimulus gets on. This would correspond to kind of a mixed stimulus. Then as, just basically because of some fluctuations, one of these will win out, or be able to maintain bursting, and the other won't be able to maintain bursting, will be inhibited by the other population. And as long as the kind of winning population keeps bursting, the other population is relatively suppressed. But over time, a hyperpolarizing current essentially builds up. This slow adaptation current builds up to the point where it can no longer maintain bursting. It will then start to die off. The other population will take over. And this is really what kind of underlies these stochastic oscillations that characterize rivalry. And you kind of get an idea about how this kind of would look like subjectively, where I basically just weighted kind of a simulated percept or an image by the normalized firing rate of each one. And you see this lovely kind of like switching behavior in the percept with a brief period of mixing around the point of the switch. Okay. So kind of with our model, before we started interpreting anything, we basically just wanted to figure out what was the dynamical regime underlying these stochastic oscillations. I mean, the two obvious hypotheses based upon like previous work is that it's either going to be a limit cycle, stable limit cycle, or it's going to be basically stochastic jumps between two fixed points whose base into the traction are modulated by this slow hyperpolarizing or adaptation current, but ultimately their stability is intact, and it's noise that drives you between fixed points. So to do this, we basically just drove the model. We set it up with asymmetric initial conditions. So you would guarantee you'd converge to one of the attractors, or one side of the ring. And then we drove it with a constant current. And basically we predicted, okay, if it's a fixed point and just noise-driven jumps between fixed points, you shouldn't get any type of oscillation, but we do. So that kind of supports our noisy limit cycle hypothesis. But then at the same time, everyone who's taking a class in dynamical systems will know that basically if you have a stable limit cycle, all stable structures will have to have an unstable structure inside them. And so we wanted to basically confirm this. And we couldn't, because of our complexity of our model, we couldn't do any of the kind of like usual analytic things that you would do. And we also couldn't make a mean field reduction. So basically we just had to work with this and use some heuristics. But the idea was basically we drove it with constant current and completely symmetric initial conditions. and found this kind of odd oscillatory structure. I'm not quite sure what it is. It might be an unstable limit cycle. But basically, we just confirmed that if we perturbed it, the trajectory would converge to the surrounding limit cycle. Whereas if we put an equal size perturbation into the trajectory when it was on that limit cycle, basically nothing happened. And so I think it's probably is some type of stable limit cycle. Okay, on to the results. So the first result was really just, we really just kind of wanted to come up with a set of benchmarks. This is probably one of the most neurobiologically detailed models of rivalry in the literature, at least that I know of. And so before we started like interpreting this kind of psychologically, we actually wanted to make sure that it made sense. So the first of these is this very broad finding that dominance duration, so the distribution of times you spend basically in each percept, will have this typical rightward skew that's really well fit by a gamma or log normal. There's a little bit of a debate about whether it actually is a gamma or log normal. I don't have a horse, like a dog in that fight. I found gamma was slightly better fitting, but not by much. But really, we just wanted to make sure that it had this characteristic rightward skew. Okay. So the next kind of probably more rigorous test of the model, or benchmark in order to compare ourselves to, was this set of kind of psychophysical laws. And I say laws in inverted commas because I know I'm among physicists. And Levelt's laws are these compact kind of propositions that summarize a really well-replicated set of findings that relate essentially the statistics of the stimulus going into each eye to basically the statistics of the corresponding percepts. And so Levelt's laws 2 and 4 are kind of now taken as the most foundational. 1 and 3 are kind of logical consequences of them. So if you've got 2 and 4, you've got the rest of them. And these have really served as benchmarks for computational models of rivalry for years. So this seems like an obvious place to start. Okay, so Levelt's second proposition is basically relates what happens when you change the strength of the stimulus going into one eye, where by strength, that you really just mean contrast. It can mean a couple of other things, but let's just say it's contrast. And I gradually decrease one eye while leaving the other eye intact. What they find basically is that the eye decreasing stimulus input, basically it only decreases very gradually in terms of its average dominance duration. Whereas the competing eye with an untouched input or untouched contrast will increase dramatically. And that's something that we reproduce in our model. The explanation of Levelt's second proposition is actually fairly baroque. It's really interesting cellularly, but I'm not going to kind of go into it because it'll take too long. But if you're interested, kind of just ask me in the Q&A maybe. Okay, the second was Levelt's fourth proposition. And this is basically the finding that if you increase the stimulus strength going into both eyes, this is now going from messing with one eye and leaving other eye fixed to increasing both eyes at the same time. What you find is as you increase the strength of the stimulus going into both eyes, basically the alternation rate, so the amount of time you spend in each attractor, basically, or each percept, decreases, right? And this actually has a really straightforward explanation. As you increase, basically, as you just blast the system with higher and higher and higher input, firing rate goes up, the accumulation of this kind of slow hyperpolarizing current increases, and so you can't maintain basically this bursting state for longer. Okay, and that's again something that we can reproduce in our model plots shown on the right. Cool. Okay, so we kind of benchmarked our model, right? We'd shown that it could reproduce these kind of like classic psychophysical laws, despite being this kind of like very rich neurobiological model. And so the next thing we want to do is we want to kind of find a result that's really kind of a simple cellular level test of this that we could hopefully test just basically by reading the relevant cellular neuroscience. And so the first thing that kind of came to us was basically just this really straightforward prediction that perceptual dominance, because it's dependent upon the maintenance of a bursting state, if we shift the EI balance in the model towards a higher excitation regime by reducing the coupling from our inhibitory neurons to our excitatory neurons, we should essentially encourage bursting and thereby also decrease the number of perceptual reversals. So increase the dominance durations, decrease reversals. And what's important about this is actually in contrast to the vast majority of mean field models in the literature, which actually predict the opposite. So if you increase inhibition, you'll decrease reversals. So here I'm showing a parameter sweep from a really great paper by Hugh Wilson, where this A parameter is basically corresponds to the inhibition in the model, or the mutual inhibition between competing populations. And what they showed was really just that as you increase A, you essentially dominant iterations blow out, even to the point where the model moves into a winner-take-all regime and it no longer oscillates. And so I mentioned before that there's been a couple of studies looking at something like Rivalry. This isn't technically Rivalry, it's actually played perception, but it'll essentially behave in exactly the same way as Rivalry. Looking at visual rivalry in mice, and they were interested in basically the effect of an MECP2 duplication as kind of a model of autism. But what's interesting about MECP2 duplications is it basically reduces the amplitude of postsynaptic inhibitory currents. And so we could also kind of look at this as a test, a really kind of qualitative test of our first prediction. And basically their findings confirmed the predictions of our model. So relative to their litter mates, MECP2 duplication mice had a reduced number of perceptual reversals, which kind of nicely reproduce this prediction of our model. Okay, so I'm going to dive into some more detailed results in a minute. Are there any questions so far?
Chris, so do mice, do they exhibit binocular rivalry?
Yes, they do.
Okay.
It's not quite binocular rivalry. It's they basically have drifting blades that move in opposite directions. And basically they track involuntary eye movements in these mice, and the involuntary eye movements track the dominant percept. And so that's how they get at this. Yeah.
Thanks.
Christopher, can you just remind me of the on the last slides about the EI balance? So you're saying if you balance the EI balance towards greater excitation, that means greater levels of bursting, and that decreases your reversals. So this, sorry, just to clarify, so this means if you are increasing excitation, you have more bursting, and therefore you maintain your perceptual state for longer. Is this what you're saying?
Yeah, exactly.
How does the bursting relate to the, it's probably a silly question, how does the bursting relate to your adaptation variable directly then? So you have more bursting, and does that mean that you can basically, you have a decreased increase in your adaptation? Or...
Yeah, this is a really good question. The answer is I don't know. So why is it that, say, shifting, another way to phrase this is, why is it that shifting EI balance decreases perceptual reversals, but just increasing the amount of drive that's going into the system will increase perceptual reversals? I think it's partially because so one of the things, these models compete, right? And they compete through inhibition. And so I think it's a combination of two things. It's basically you are in a bursting state for longer, but the competing population isn't able to take off and then inhibit the currently winning population as easily.
I see. So it's kind of over. It's because you could definitely imagine the opposite. You know, it's bursting so much.
Yeah, exactly.
And therefore its adaptation goes up, and therefore it switches quicker, which I can understand why there's like confusion or like kind of opposite predictions. So yeah, so it's kind of it's winning so much effectively that the inhibition can't, it can't inhibit. Okay, cool. Thanks.
Yeah. Okay. So
before
all
you
these
keep going, Chris, I just wanted to double click on the question you were asking, Claire, about the rivalry in mice. It's actually an interesting reason that it's a little harder to evoke in mice, which is that they have less of an overlapping binocular visual field than other.
Their eyes are like pointing that way.
Right. They're kind of communing a little bit. And so there's something like a 10-degree overlap in the middle, as opposed to our ridiculous 130 or whatever degree we have overlap we have. So it's harder to do, but it's possible. And then you have to come up with these tricky things that can kind of get at the phenomena indirectly. Yeah, it's a good question. I thought it was an interesting explanation for it.
Yeah. I mean, I wonder how they define what's a voluntary and involuntary eye movement in a mouse, but it's interesting. I can see how it would be hard to get at. It's cool.
Let's not go all Sapolsky on this talk, Claire. Okay, it's like let free will stay alone.
Oh, don't worry. I've got a lot more questions.
All right, Chris. Sorry for interrupting you.
Okay. So far, I really just talked about some predictions that I think most spiking models would make. It didn't really involve anything to do that was kind of novel about our model. So the next thing we want to do is basically to kind of recreate some of these perturbations that they did in Matthew Larkham's group in this threshold detection paradigm, but in our rivalry context. And so I'm thinking about this before I go into what we did and the results, we'll kind of think about this more abstractly. If we optogenetically or simulate an optogenetic kind of excitation of one population, what we're doing there is we're basically shifting ourselves rightward on this firing rate function, right? We're increasing the amount of basal drive that's going into the soma when we're leaving the other one intact. So basically, if we think about Levelt's second proposition, what we should do is this will basically, we'll see a large jump in that guy relative to the other one, right? And then if we have the opposite, if we have a pharmacological inhibition of one population, which again, we just simulate with a constant current, and leave the other one intact, we should basically see a flip. So the inhibited one should decrease fairly slowly, but the other one should have this large jump. And that was actually basically exactly what we found. So if we basically added a constant excitatory drive to one side of the ring, this was basically representing the input in from one eye, we see this large jump in the dominance duration of that percept. And this could basically be compensated for by decreasing the contrast of the stimulus, or basically here it's not contrast, here it's the frequency of our Poisson drive, decreasing the strength of our input stepwise. And we see the opposite finding, or the reverse finding, in the case of inhibition. And I actually hope this is something that can be tested. I have had a few chats with some people who work in kind of systems neuro, in particular, someone who was doing neuropixels in primates. And so this is a bit of a wild prediction. You definitely can't do optogenetics in primates, but you might be able to do some pharmacological manipulations to get at this. And I think that's kind of where I hope this goes. Okay, so I started by kind of promising, or with the goal of this talk was to kind of link from these psychological level functions, right? That we study in cognitive neuroscience to these kind of cellular level things. But so far, I've just talked really about cellular neurobiology. And of course, in a biophysical model like this, actually making contact with cognitive constructs that are of interest to cognitive scientists, it's really hard. You can't really do it all that well. But what we did was kind of somewhat the opposite. We didn't try and build it in. We basically took, given our kind of macro scale structure, we then went and kind of read the relevant kind of cellular neuroscience and looked at all of the functions that these things have been implicated in, and those corresponding functions and their role in rivalry. And so attention, for example, has been linked to basically the selective boosting of cortical responses through the matrix thalamus, basically selectively boosting feed-forward input. So if you inhibit the pulvinar, which has a whole bunch of these like matrix-rich cells, you'll basically diminish cortical responses to task-relevant stimuli. In rodents, you can get the opposite result. So if you excite the medial dorsal nuclei, which again is rich in these matrix-like cells, you'll increase cortical excitability. And then expectation is kind of something I think everyone here who's probably read the Carl Friston paper is kind of aware that basically expectation has been linked to these feedback projections that terminate in superficial layers and are sent from these deep layers. And so we kind of hypothesized that basically if we just add an external source of Poisson drive to our apical dendrites, we should be able to reproduce some of the effects of expectation. Okay, so we basically picked attention paradigm in rivalry. This is actually something, so David Elias and Franz Vertassian actually in the psych department here at UCID. Basically, they have this paradigm where they coincidentally manipulate attention and rivalry at the same time. And the way they do this is by adding this kind of motion coherence task in the background of the rivalry stimuli. But they titrate the difficulty of this between participants so that their attention is basically, they always have to have a certain level of behavioral accuracy. And what they found was essentially that removing attention was essentially equivalent to a stepwise decrease in the contrast with the stimulus. I've actually noticed that I think I kind of butchered the x-axis on this plot. So the 47 and the 23 should be reversed. So sorry about that. But anyway, basically what you get is that if you look at the difference between the attended stimulus and the attend away condition, basically it's equivalent to a stepwise decrease in stimulus contrast. And this can kind of be really simply explained, right? So if we prime our matrix, if we prime the thalamus, or what attention's doing is it's priming the thalamus. So when it then receives this projection from the cortical ring, primes the thalamus, it will then have a higher firing rate. So it'll then project back up and contribute to the higher firing rate of the apical drive. We're essentially moving ourselves rightward on this, firing rate plot at the single neuron level, right? But leaving everything else, including inhibition, intact. And this is essentially moving rightward, but in terms of Levelt's fourth proposition, this is essentially moving rightward on this graph. So we would expect actually that if you increase attention, you go from this lower plot to this higher plot, you should increase the number of reversals. And that's what they found. And that's also what we found in our model. So we basically swept the parameter, the coupling parameter for our thalamus to our dendritic population. I've just picked two of the ones here, two values here, but we do see this same qualitative effect where we see a stepwise increase in the number of perceptual reversals. And that is equivalent to changes in stimulus strength. Okay, so this is the last result. We wanted to kind of look at something a little bit more sophisticated, so we chose expectation. And there's this lovely study by Yael Pinto, senior authors in Nielsen, where they didn't actually look, it's actually not classic binocular rivalry, it's a variant of visual rivalry called continuous flash suppression, where basically to one eye, you'll receive this very high contrast mask, and to the other eye, they'll have a very low contrast stimulus. Here it's a face or a house or something like that. And they'll gradually linearly sweep, decrease the contrast of the mask and increase the contrast of the face going to the other eye. And at some point, the face will break, the stimulus will break through, and the participant and the person's experience will basically switch from this experience of this mask to the experience of the face. And basically what they did was they then added a predictive cue at the start of each trial. It had basically a 75% validity. And what they found was basically that this valid expectation relative to invalid or neutral expectations sped up breakthrough times. That was the time it took for the stimulus to kind of like break through into perception to be able to accurately report it. And they have a whole bunch of kind of like controls to make sure this isn't just a criterion level problem. But I won't go into that. So basically the way we reproduced this in the model was just by targeting a select part of the ring with an external source of Poisson drive, or that was basically also receiving some coincident basal drive, or targeting a different part of the ring. And so I think fairly trivially, it will just follow that if you basically hit a part of the ring where there's almost no basal drive, right, there'll be some depolarization of the dendrites that don't really do much. And that's essentially exactly what we found. So you get this speeding when you basically add an external source of Poisson drive to the ring. And so I think this basically just fairly trivially follows from the structure of the model. But that's kind of nice in a way, because we didn't build the model with kind of the intention to model expectations. Okay, so just to wrap up, what I've shown is that we have a cellular level model kind of consisting of this minimal thalamocortical circuit that's been kind of implicated in systems and cellular neuroscience as a potential kind of basis of conscious perception. We've shown that this kind of generalizes to this much more complicated task that's typically used in like cognitive neuroscience, namely visual rivalry. We've shown that it's consistent with psychovisual benchmarks, and it generates kind of some novel predictions across levels of analysis. So cellular, mesoscale neuroanatomy, human psychophysics. And regardless of whether any of this is kind of, it turns out to be correct, I'd be surprised if much of it did. What I really want people to take away is just that this is kind of a useful thing to do. Even if you're interested in something kind of as relatively high level as binocular rivalry, you can actually learn a lot by reading the relevant kind of cellular neuroscience of perception, or whatever it is. Maybe it's the cellular neuroscience of decision making. Okay, so with that, I'll thank you for your time. And also, yes, please see the preprint.
Great stuff, Chris. It's lovely to see it kind of laid out with all of the background and the thought that went into the project. And I think also my mind started wandering halfway through towards the kinds of things that we can do next as well, because there's just so many other questions that pop out along the way? What about this other thalamic population? What if you wire them together? What if you modulate them? What if you get them to do a different type of task? There's so many things you can do with this that get me really excited. So really lovely work. I'm curious whether people in the group have questions, parts that were of interest, parts that didn't make as much sense, parts I'd like to have elaborated on. You know, we're in a safe space here. So if you have any questions, please feel free to jump in and ask.
I'll have a swing, Chris. So you made a point of referring to Hugh Wilson's work on Rivalry and like the neural mass model, right? But like, I haven't read that paper, but presumably there's like quite a stark difference between what your model has incorporated and what they have in their description, both mathematically and neurobiologically. So like,
Yeah.
You said that, you know, you've seen this inverse relationship in terms of the way the dominance durations scale as a function of inhibition. Like, do you have a concrete intuition about within your model, what were the essential components? Because presumably like you've added in something like adaptation as well as a spatial component. Whereas if they've done a mean field setup, I don't know what spatial component they've set up there, but you have this distribution of connectivities along your ring. And as you're scaling contrast, like all I'm saying is it a fair comparison to say, you know, we've both increased inhibition, but because you do yours in a spatial sense and you have this addition of adaptation, that's driving the difference? Like, yeah, anyway,
Yes.
Do you have a feel for it?
No, that's a really good question. Very fair question too. So the Hugh Wilson model, they do have adaptation. It's this like 4 variable mass where it's basically 2 excitatory masses, mutually inhibitory and each population has an adaptation variable. I think the key difference between the models is that basically for them, the gain of these neurons, of these masses, does not depend upon a thalamic projection or something that's at least somewhat external to the population. That's my feeling of the difference. I think if we basically made bursting depend upon the gain of a population, right? I think we could probably reproduce this in a mass.
So like in your model, when you're scaling contrast, like what does that actually represent? Like you have these Gaussian projections to 2 locations on the ring that represent the 2 stimulus. And presumably you turn up contrast, it scales the amplitude. But with that distribution, scaling the amplitude also scales the spatial extent of the stimulus on the ring, because there's more energy to in the extremes of that distribution as you raise the floor.
Because our model approximates, I'm sure it does, but I don't think the effect's all that big. Like just like this is just my intuition now. I haven't actually checked. That's something I should check. Basically, the spatial extent of the population, like in these raster plots. Okay, we can go. I think it was on this last slide. Whoops. Yeah, in these raster plots, the spatial extent. Okay, the spatial extent of each of these populations doesn't actually change all that much as a function of stimulus input. And I think that's because the thing is so heavily inhibition damped, basically. You have this very tight local inhibition, but if you kind of move more than maybe 10 neurons either side of the center of that population, it's really heavily damped. So my intuition is it wouldn't do much, but I should check, absolutely. And I guess one thing I kind of wonder about is the extent to which we should probably try and build. We can't get like an analytic reduction to a mean field, right? But just the extent to which maybe we should try and build this into a rate model just to see if kind of some of the explanations that we've come up with generalize to this kind of less idiosyncratic setting.
No, yeah, for sure. Cool.
Hey Chris, I also had a question. It's kind of like more just extending to like other like phenomenon. So maybe a bit more niche, like thinking about like cross-eyed in that like from what I've like kind of like just heard or just read briefly, it's like each sensory information you get from each eye is independent of each other. So it's kind of similar to what you see at the start of your model where you see both the red and green before then you get the dominant and then the adaptation. So would you say that if someone has like is able to do like the cross-eyed vision where they can overlap 2 independent stimuli, is that more like they just lack like some the adaptation part where they're stuck at the start of like what your model shows, and then there's no like dominance or adaptation? Like, I'm wondering like if you have any ideas about that.
Yeah, it's a really good question. So I should say, actually, basically Rivalry, when I started this project, I built my own little Rivalry setup, and I spent a lot of time just kind of like sitting and like lying on my bed, just kind of like thinking about what could possibly be going on here. But I also basically set it up on most of my extended family, including my partner's mum, whose eyesight sucks. And she basically doesn't experience Rivalry because of that. If you have any, turns out this is actually a really well-known thing to people in psychophysics. If you have any type of like asymmetry between your eyes, basically binocular Rivalry doesn't work anymore. So that's the first thing to say. But the second thing to say is that one of the things that makes Rivalry work is that you have to have completely independent inputs into each eye, right? Like there's no overlap. Whereas even if you're going cross-eyed and you're just having like one stimulus directly in front of you, there'll actually be quite a bit of overlap in the visual field going into each eye. And so I suspect that's probably what's going on.
All right, thanks.
I have a question. Oh, you go, Ben.
Well, one boring one. If you really wanted to compute the equilibria, could you? Like numerically?
Oh, numerically. So
how many coupled ODEs have you got here? If you set all the derivatives to 0, is possible, no?
So I think the issue is that the equilibria, there would be a trivial equilibria in the resting state, but the non-trivial equilibria would be something like the steady state firing rate, which, because they are non-continuous, right? They're basically these, they take off, these models get input, they take off to infinity in a quadratic, and then they cut it off in a reset. There wouldn't be a well-defined fixed point in this model for anything but the resting state.
So they're actually, you would think of them more as metastable.
Yeah. So I think it's a limit cycle personally. That's my going hypothesis. Is that what's going on?
But you could look at, limits on that time scale of the slow process.
Yep.
Set that time scale to the appropriate limit, and then look for equilibria in that regime. Or I think if you really wanted to, there could be things you could do, but maybe because you mentioned, we couldn't find, whether there was an unstable state there, or maybe it wouldn't add much. But I think if you wanted to go there, there are probably ways you could.
I might send you an email after this, actually, because that was something, at least maybe not for this project, but something I'm very interested in, is trying to figure out how we can get kind of more of the lovely things are interesting. Yeah.
There are people in maths whose whole career is this, right? Numerically computing bifurcation diagrams of really complex ODE structures, and like that's just what they do.
Yeah.
It's not me, but I could, if that's where you wanted to go, I could point you to those.
Yeah, please. And to answer your earlier question, it's about like 400 coupled ODEs.
Yeah. I don't know. It seems large, but I think it's doable. Numerically, when you're just finding equilibria. But and I don't know how indulgent it is to ask a double when Claire's waiting, but
no, go ahead. Please.
Well, it kind of hooked me with all the background. Like that's like your opening 5 or so slides were so fascinating, in being such an emergent piece of science that kind of encroaches on things that were conventionally taboo to talk about in science. And then we kind of got to the end. I guess if you had another 5 slides to give your vision again of how this would fit into, that literature that you opened at the start, right? Because you've got, IIT, kind of philosophically and conceptually different ways of getting at the problem of consciousness. And now you've shown, look, you can actually make quite concrete physiological predictions and model them and get some insights. Like, where would this lead to its own kind of independent stream, or do you see it interfacing with some of these other theories of consciousness? I guess I was interested in your thoughts on it.
Yes, this is something that Jana Rue has really kind of led the charge on. So they had a nice paper in Trends in Cognitive Science, kind of speculating about the links between something like what they're calling dendritic integration theory, which is really these like the importance of this layer 5B kind of recurrent loop with matrix thalamus. And what they say is basically they think that it captures most of the flavor of IIT and global workspace theory. And I should say Brandon actually has some work where they basically just have a sheet of these neurons, and they basically sweep a whole bunch of parameters. And what they find is that at the point where there's kind of the empirically plausible point in the parameter space, there's a maximization of both kind of like phi star and the participation coefficient and a minimal participation coefficient. So you see a breakdown in modularity, which is kind of what you would think of as kind of a signature of global broadcasting. So that's the first thing to say. I think the second thing to say is I would actually really love to do, here we really just kind of focused on like behavioral signatures. We actually didn't do much in terms of neuronal signatures. But I think the work that we're going to be doing in the future is we're going to be trying to basically build really big populations of these guys and seeing if we can reproduce some kind of like EEG signatures. And hopefully linking to some of the more sophisticated paradigms that people who are interested in global workspace theory and IoT are working on. And I'm as best as I can trying to stay like theory neutral. I think I'm way too young to have a theory. But I just really like this thalamocortical circuit and modeling.
For me, I like it because there's that question of, does the algorithm have to be embedded, or, could you abstract it out? And people just think computationally, what is the brain doing? What aspects of the computation might have subjective experience? When you model it as ODs, you're explicitly saying it's nothing about I can abstract it out as an algorithm, right? And that's capturing something about it. And then you get all these people talking about, well, what's different about cortical computation to, they're just really thinking of it as an input-output algorithm and, Turing completeness, all these sorts of ways of thinking about the brain as an algorithmic machine. And when you write it like in a set of 400 equations, that's explicitly a substrate-independent algorithm. And so you're moving it in that, or at least interfacing with those ways of thinking about the brain as a computation.
Yeah, absolutely. I think what I would say is what we're modeling is probably a necessary condition for conscious perception. If all of this dendritic integration stuff is correct, I think what we've done is we've modeled a necessary condition. I think the hard thing is thinking about the necessary and sufficient conditions. And my personal suspicion is that there's going to be a whole bunch of like really bizarre background conditions to do with us being like a living system. That's going to be really hard to build in this kind of more algorithmic sense.
Great work. Great work. Thanks, Chris.
Thanks.
I'll let you finish your having a sip. That was a really awesome talk, Chris. Thank you. Yeah, like Ben, I really love the kind of lead in and the background and kind of couching it within I don't know, these sort of different kind of streams of work that you pulled together really nicely. And I love the approach you're taking and thinking about a sort of, how to kind of pull together these kind of cross-species findings. So I might be kind of leading, I think I'm sort of leading you down the garden path to a slightly bigger question. But my first one is that I guess I was sort of thinking, I don't know that much about the binocular rivalry literature, but I know it's a task that's sort of done to death in psychophysics, etc. And I guess those sort of cognitive science and psychophysics tasks, always slightly terrify me because of the I guess because of the idiosyncrasy of their findings, in that you basically have to be a 19-year-old undergrad locked in a padded room and sitting through about 800 trials for these effects to work. And then when you change, you tweak a slight parameter, they don't work. So yeah, but I guess the one of the things that sort of I just found myself wondering, through your background is what's happening in monkeys? Surely this is something that is binocular rivalry is surely done to death in monkeys. And I guess I just wondered, is there a bridge there where people have actually done this in a system where the rivalry is probably a lot similar, given how they're built, and actually have probably done some sensible sort of neural recordings and stuff. And I guess I wondered, is there anything there that kind of speaks to your to this kind of path that you led us down?
Yeah, absolutely is the answer. So non-human primates also obey Level's laws, or the findings also seem to obey all of these same kind of classic psychophysical propositions, which is kind of encouraging, right? I think that shows that we're not studying something that will disappear the moment we go from like studying undergrads in Sydney to undergraduates in New Zealand or something like that. And yes, they do a lot of neuronal recordings. Typically, they'll look at LFPs or single cells. And I think the big picture thing is it's typically there'll be synchrony in the gamma band for the dominant percept. I was like I think I'm very excited for that because I think, or for future work in this area, really because neuropixels can now be done in primates. But it was hard for me to kind of tell what was going on from like an LFP to a lot of the kind of layer specific structures that I was interested in.
Yeah, it's not making contact with the level of analysis that you're working at. Yeah. Yeah,
exactly. But I am feeling really good about, or really optimistic about kind of the future of that. I mean, the white paper for neuropixels and primates came out this year. And I actually had a chat with someone at Maths in the Brain who's going to be doing neural pixels and non-human primates and binocular rivalry.
Yeah, nice.
Yeah.
Yeah, that's really cool. And I guess, that it was kind of like my bias in that question was, having sort of spent a lot of time trying to link, task, behavior and neural correlates across species. I think I'm especially in the last sort of 12 months, I've just there's so many things where I'm just constantly floored about the species differences, right? That I keep coming across. And we were, famously really well schooled by Amy Arnston herself in one of the reviews she gave us, that we'd kind of drawn we basically made all these kind of claims based on, rodent literature. And, it's actually so different if you kind of go across rodents to non-human primates. And I guess, that's something that I've been sort of coming across lately, and it's more from a neurogenergic perspective, but actually trying to sort of thinking about kind of early sensory processing and, where you actually see this sort of, really important gating effect where subthreshold stimuli is then kind of boosted to sort of perception and stuff. And I do think, in some ways that does make contact with this literature. But one particular finding that really stood out for me there, just for example, is I went down this rabbit hole of this sort of locus coeruleus direct projections to the dorsal lateral nucleus that explained all these really interesting effects about about this gating effect, right? And then you find out that those projections are actually completely sparse in non-human primates. So this kind of explanation of what's going on in rodents, it doesn't actually hold. And I guess the reason, it's a bit sort of rambling, but I guess the reason I'm sort of bringing that up is that I do think it's something that is really important to think about when you're dealing with stuff to do with kind of early and then even slightly higher level sensory perception, because I think this is where there are so many baked in species particularities, because sensory it's like the gateway to how we function in the world. And I guess it was more kind of just trying to sort of encourage you to, when you sort of think about drawing these extrapolations, particular for quite specific cellular architecture and projections and stuff, to really try to see if it does carry if the neuronal architecture and the layers and distributions of cells and layers and stuff carry across in a really robust way between at least rodents and monkeys.
Yeah, absolutely. That's something I'm actually a little bit concerned about, worried about.
And I mean, it shouldn't disturb you at all, but it's more just like, I would encourage you to, like, when you sort of think, oh, this is awesome, and it explains in rodents, just have a little scratch around and find if somebody's even looked, if that projection and that kind of, layer specificity and things, if that pattern actually exists in monkeys. And I guess it's just, based on some few times when, particularly with early sensory and perception type stuff, that I've been quite sort of amazed at the differences.
Yeah, that is something I should really dig into. Yeah, I don't really have anything more intelligent to say other than that's a really good idea, and I should definitely do
Yeah,
these
no,
kind
it
of
was more
checks.
of a, it was certainly more of a comment than a question.
Yeah. No, I absolutely agree.
Yeah, it was mainly just kind of based on my own getting into a complete, getting really excited that I had an explanation for something, and then for something we'd found in humans, and then realizing, oh yeah, hashtag, in mice only.
Yeah.
I wouldn't be afraid of this though, Chris. This is
No, I'm not.
exciting.
No, totally.
Place to go wander around and think. And one thing we don't talk about enough, and Paul Chizek brings this up a lot, which is that if you really care about changes in the nervous system across phylogeny, and you start to think about what kind of access we have to the kind of big bristling bush of natural selection that's out there, we have these tiny little pockmarked samples of this incredibly complex space, and we're just missing so much information. And what we end up doing is we say something like, well, this particular Cree line of mouse that I was given has this particular feature. That must be characteristic of all mice. And therefore, it must be this particular feature that we're trying to find, is either, trustworthy or not. But if you go and talk to mice researchers about their particular strains of mice, and you give them a couple beers, they start talking to you about how weird these particular strains are. They're like, oh my God, this one particular type of mouse is so strange. It doesn't do this, this, and this, but it totally does this other thing. Or if you talk to monkey researchers, they say, oh, figure 1 was the good monkey, figure 2 was the bad monkey that was always in a bad mood and never.
They've always got the wildly different personalities. They're like angry teenagers when you talk to monkey researchers who look after them every day.
100%, right? And so I know this comment is about as rambling as Claire's one was, but it's to get at a point as well, which is that there is little stability at all in the literature. And so I think what we ought to do is keep as much of an awareness of what we can conclude from science, as opposed to like looking for these, points that just solve everything. I just don't think that answers that we're going to get are going to be these like forked lightning, evolution by natural selection type answers. They're going to be, in my particular model organism, here's a pretty decent explanation for what was going on. Now, is that related to other animals solve that? Or is that related to the way that this thing works in other species? Is a completely different question. And you shouldn't expect yourself to solve that in your project. I think that's kind of like, you can kind of like unburden yourself from it. And then you can go looking for stability and selectivity. And, I don't know. I think just an appreciation of the complexity of the animal world is enough.
Yeah, and that's all I meant by that comment. It wasn't meant to a deterrent. If anything, it's actually like a beautiful invitation to scrounge around the literature and actually look for, read some monkey literature and be like, oh, is this the same? Has someone found it? Oh, cool. And if it's different, why would it be different? And then what does it mean if it's different? It wasn't by any means a deterrent, but it was basically just a kind of encouragement to kind of dig around and think about the kind of whether your applications are sort of wide or more kind of narrow to the model that you've built and the model organism.
Yeah, absolutely.